Today we will discuss about a therapeutic approach that is the conjugate of nanodrug and bacteria that helps in reduction of this PDAC, immunosuppression and tumor proliferation properties. So we will first understand how the PDAC promotes this tumor proliferation and immunosuppression. So tumor releases this oncogenic collagen that interacts with tumor cells, integrins alpha 3 beta 1, and also this tumor microenvironment has this anti-PDL1 antibodies that binds to these PDL1 receptors so that the immune checkpoints therapy will be ineffective. So because of these two things, what will happen? Because of the binding of this anti-PDL1 ligand, anti-PDL1 antibodies to this PDL1 ligand, the immune checkpoint therapies will be ineffective, that I have just said. And when these oncogenic collagens interact with this integrin alpha 3 beta 1, it will trigger a intracellular signaling pathways that is composed of this FAK, AKT, MAPK activations, and this leads to what this triggers the cell proliferations and survival. This helps the tumor cells in survivals. At the same time, this FAK activation of this FAK will promote immunosuppressions that will prevent the tumor cells from immune attack by the host immune system. So in order to counter these things, the approach is that they will use a nanodrug that is incorporated in a bacteria, uh, and for example, and that nanodrug contains protein A and protein B. Protein A is basically a collagenase, and protein B is this anti-PDL1. Anti-PDL1. So what will happen is that, and this is incorporated in a bacteria, that is E. coli, and E. coli has the kind of E. coli bacteria that they used has the specific property of hypoxia tropism, that is it will go into the hypoxia environment easily. So because of this property, the E. coli bacteria can enter into the hypoxia ECM environment of the tumor cells, and where it releases collagenase and anti-PDL1 and and this anti-PDL1 antibodies can compete with this anti-PDL1 that is present in the tumor microenvironment to inhibit these PDL1 ligands. So because of the competition of these two antibodies, the efficiency of this autoimmune response increases because of the increase in the amount of anti-PDL1 ligands due to the nanodrug bacteria conjugates, and this will we can say that this will activate the this immune checkpoint therapies response. And the other things, that is collagenase, it will uh, digest this oncogenic collagen. It will digest the oncogenic collagens, and that will leads to the inhibition of this immunosuppression and cell proliferations. And once the immunosuppressions get reduced and cell proliferation is also reduced, the immune system can come and act against these tumor cells. And this is the basic reactions that is usually occurred when the nanodrug bacteria conjugates is used. It is protein A and protein B when it's cross-linked in a bacteria or biochemicals conjugations occurs, and then it will engineer or engineered to the tumor microenvironment area of the pancreatic tumors, where it will act and target on two different ways. First one, it will increase the efficiency of immune checkpoint therapy, and second one, it will reduce immunosuppression and cell proliferations. So this is one of the novel therapeutic approach that can be used to diagnose the aggressive pancreatic adenocarcinoma. And I hope you got a basic idea about how this therapeutic approach works, And if you need to understand more about this idea, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.